Okay, so hi everyone. It's my pleasure to have Roger coming in to give a session on moths. So Roger Kendrick, as everyone knows commonly and popularly as HK Moths on iNaturalist, I'm pretty much sure almost everyone has have got some of the other IDs from him at some point. So Roger, as everyone knows, he is into moths. He founded and he's coordinated the biennial Asian Lepidoptera Conservation Symposium series. He works in a lot of events like the National Moth Week and is part of the National Moth Week Science Advisory Board, etc. for Hong Kong. He manages a lot of groups, including online projects like INAT for Hong Kong, Singapore, Malaysia, India, Sri Lanka, Vietnam, Cambodia, you name it. Basically, he works on mods pan Asia, and I'm really happy that he accepted our invitation and he's here to give a session on mods. So we'll hopefully everyone makes the best out of it. And yeah, over to you, Roger. Thank you.
Thank you, Ashwin. Pleasure to see lots and lots of people joining now. Of course, this is being recorded so you can catch up with what I'm saying later. And I will say I'm going to be able to say things only once. So please listen in, hear what you say one time around. If you can't digest it, then go back to the video that will be posted by Asian Nature Challenge on their YouTube channel. And maybe even on their WhatsApp channel. I'm not too sure about that, but Ashwin can brief us later. Right. We are seriously time challenged tonight, or this morning, wherever you are in the world. Let's get cracking. I think I can figure out how I'm going to operate stuff. So we are going to be looking at how to contribute moth data to iNaturalist for Asia Nature Challenge specifically. I'm going to take it slightly broader than that, give you an idea of diversity of moths, so give you some idea of what we're up against here, how to go and record them, what kind of weather conditions are important, a little bit on moth photography for recording. I'm going to go into a little bit of depth about what an observation actually is. INAT kind of jumps the gun a little bit on some of this and simplifies it rather, but we'll go into a bit more depth. Then I'll look more specifically at the main moth families found in the Oriental region, and we'll link back to the diversity there shortly. And finally, we'll wrap up with actually how to keep the records on INAT. So although this is about iNaturalist, not very much mentioned until we get towards the end, which is really kind of the take-home point. So, diversity. Before we roll specifically on families, I would love to introduce to you the classification name endings for the main ranks I will be talking about. So here we go. At superfamily level, and superfamily is kind of big groups of families, they end O-I-D-E-A. Now, I pronounce that oidea. Somebody else might pronounce it a bit differently, but bear with me. A superfamily that is big in this part of the world is the Gallicioidea. Note the suffix is highlighted for the name. The prefix, which is derived from the family that is the type family for that superfamily, is then appended in front of the suffix. So for family, the suffix is IDAE. And in the family Galicidae, watch out, because you have a stem that ends with IA for the genus Gallicia. You drop the A, but you double up the I, so you have Gallicae and then IDAE at the end. One of the subfamilies of the Galicidae is the Anacampsinae, so INAE rather than IDAE. Tribes below subfamily end in INI. Now, how you differentiate INI from INAE in classics, I do not know. So to me, Anacampsinae and Anacampsinae could be the tribe or the subfamily, but when it's written INI, It's the tribe. Now, genus and genera, gen for singular or plural. Then subgenus, subgen is an abbreviation. Species, SP for a single species, SPP for many species. Likewise, subspecies, SSP and SSPP for the plural. Right, so that's name endings. That's actually really important because it tells you immediately what kind of hierarchical level or rank we're talking about. Now, diversity at the family level, these, I'm not going to go through them now, are the main families found in the Oriental region by numbers. There are some families I've not put on this list. I could look at the Lasiocampidae for a start. Some of the microfamilies, Adelidae, for example, are not on this list, but they're present in this part of the world. Now, the size of the problem? Well, there are something like 160,000 species of moth described globally. But that's what's been described. It's thought, based on what we keep finding and what is known to be undescribed, particularly in Australia, is that we could be looking at over 350,000 species of moth. That puts it as part of Lepidoptera, the scaly-winged insects, as by far the majority. Something like 25,000 butterfly species, the Papilionoidea. All the other superfamilies are moths. Now, how to attract moths? Well, the old moths to the flame is the adage, and it's perfectly true. What your flame is up to you to choose, but try and find something that has ultraviolet light in it. Ultraviolet light has a wavelength, at least for UVA, the relatively safe ultraviolet, of about 360 to 400 nanometers. That's a shorter wavelength than we can see. So what I've put in the text is what basically the moths can interpret that wavelength as. It was basically this kind of deep, deep violet blue. And light sources that we can use to produce mercury vapor wavelengths, to produce ultraviolet wavelengths. Good grief, I need a shot of something in the arm. Mercury vapor lamps, these are used on a commercial basis and for recording as the primary modus operandi for bringing in moths, in combination either with a bucket style trap or with a sheet. Other sources of UV are actinic lights. This is an actinic light in operation. It shines this blue because it gives off blue as well as UV. Or you can use light emitting diodes like this lot. This is a rig I've built myself. The UV components, the two diodes that appear to be very, very dark blue and not emitting any light at all because you can't see the UV. The other lights are green, a pale cyan and white and blue. So blue, green, ultraviolet, cyan, white, white, another cyan and ultraviolet. And that combination is 12 watts total output. But the key thing here is 6 watts of that, these two ultraviolet. Basically, the white is to help us see what's around. The old tungsten lights before we had the more modern, relatively energy efficient fluorescent tubes and the like are used still in light trap designs, particularly the Rothamsted traps, where in the UK we've had a series of light operated traps running since the 1950s, and to maintain the continuity of the method, they still use tungsten lights. I don't know how long we'll be able to get tungsten lights though. Other sources, halogen lights, bright white, particularly used in stadia, sports stadia. Those of you that followed the Olympics in, I think it was Australia, Sydney Olympics 2008 maybe, might recall that the floodlights attracted a passing migration of bogong moths, but that's an aside. Right. Lights are put in combination with traps of various designs. Easiest to see is combine your light with a sheet. Downside, you've got to be at the sheet all the time. So that's great if you're wanting to be active by the sheet all the time. It's good in the tropics because light sources bring in not just moths, but big beetles and all sorts of other things, praying mantis, for example, snakes, frogs, you name it, they come, bats even. And if you have a trap, getting any of those other taxa, shall we say, involved can be rather detrimental to your survey. So alternatively, box-style traps or bucket-style traps can be used in combination with your light. Now, a box-style trap, basically Heath-style or Skinner-style, after Bernard Skinner and John Heath, prominent UK lepidopterists in the 1950s through about 2000, designed these particular traps. These are collapsible. They basically is a box that unfolds. There's no base, and then you put your folded up out box onto a sheet and put the egg trays inside. There is a clear perspex top that slots in on both sides. The moths go in through the gap underneath the bar that holds the light and can get back out again. Better retention is from bucket style traps that have a funnel into the trap. So this is a circular funnel that goes down into the trap. The moths drop into the trap from the center. Again, a perspex collar. Trap is filled with egg trays. Egg trays, by the way, mimic tree bark. So moths nestle in these feeling reasonably comfortable and basically shut down because it's quite light in the environment. Moths generally, at least the nocturnal ones, switch off their night vision, settle down for a little stint in the moth trap until they're let loose when you go look inside. Alternatives to lights, you can use nets, sweep net in grasslands, for example. This has got a reinforced net rim for sweeping through tall grassland or thorny vegetation, things like lantana scrub, acacia scrub. For more open field situation or aerial netting, you can use a hand net. This has no reinforcement around the rim. Different sorts of nets. You can use a Malaise trap, which is basically a flight interception trap. This is kitted out with a bottle at the top containing alcohol to capture and kill all the insects that fly into the trap, then walk up, walk along and into the bottle. Downside of using particularly liquid killing agents is that for scales on butterflies and moths, you end up scaleless samples. It's just not wonderful. You can use baits. Popular ones include sugar. Come to that in a moment. Wine roping. So you dip your unbleached rope into wine, let it saturate, and then drape your rope over vegetation. Sugar is, in effect, fermented fruit blended in with a good dark bitter or stout, brought to a gentle simmer. Loads of sugar. Demerara sugar works really well, or good dark brown sugar. Bring the whole thing to a simmer. Add in something like pear drops amyl acetate or a tot of whiskey, brandy, cognac. That helps lift the scent of the bait. Plaster the bait onto a tree trunk, a fence post, or drape a cloth into the solution and then put that over a branch. And you end up with lots of species quite often coming to the bait. This is particularly effective when you have, in the UK, for example, trees are in flower in spring. Elsewhere in the world, you have to be a bit careful not to bring in other animals. Ants are particularly partial to baits. Snails are particularly partial to baits. I have no idea whether larger animals like bait, but I could imagine bears being rather attracted to bait because it's not so different from honey. And you know about Paddington Bear and honey, Winnie the Pooh and honey. Bears like honey, so probably they're going to like the bait too. Anyway, you're doing mothing with bears around, you have been warned. What else? Well, natural fruit, of course, acts as a bait. We have a number of moths that we call fruit-piercing moths for a reason. They actually pierce the fruit skin with their very stout horstellum, come back to that word later, and get pretty drunk if the fruit started rotting. Makes them easy to see. The last kind of main bait I'm going to talk about is synthetic. We can manufacture a long string benzene compounds that mimic natural pheromones, scents that are emitted by moths. This is the reason they got their antennae looking feather-like. And using these synthetic pheromones, we can monitor or capture particular species. And the cecity and the tortricity in particular, well documented and to be recorded by pheromones. Now, whether the weather is good or whether the weather is not. For light trapping, forget windy weather. Ain't going to work. So if you know your Beaufort wind scale, anything above a Beaufort force 4 on average, forget it. Not worth it. Moths do not like strong wind. If there's heavy rain, not optimal. Moths do fly in heavy rain, and I've seen even big Actius species, the moon moths, flying in big thunderstorms in Hong Kong. But on the whole, not optimal. Light rain, on the other hand, misty and warm conditions. Oh, get out there, go mothing. Absolutely. Can be really, really stunning mothing. You can often find in a misty mountaintop, put your trap out, it will be inundated if there's not much wind, but there's a mist or a fog. And now in Hong Kong, temperature is also important, as it is anywhere in the world, for insect activity. Moths and butterflies are both thermophilic in their way to live. They like heat. They have to warm themselves up. So in tropics, where it's a bit warmer than it is in the UK, you won't see moths vibrating their wings very often because vibrating the wing muscles warms the wing muscles up to get an optimal temperature. They operate at something like 40 Celsius. If you have warm nights, not too cold, not too hot, then that seems optimal. And in the tropics, you get good temperatures at higher elevations, 1,000, 2,000 meters. In the UK, if you're operating at that kind of elevation, you're going to not see very many moths. You need to be down at low elevations to get warm temperatures. Even here in Hong Kong, when we have our dry season, it gets cold, January, February, and drops down below 10 degrees C. We don't see much happening. And one can say much the same for looking for larvae. Forget looking in heavy rain. It's A, uncomfortable for us, but even more so for the larvae. And they get washed away, quite literally. So wasting your time. After rains and in calm weather, definitely. Not heavy rain. The same can be said for baiting. Sugar, pheromones, however you want to do it. Calmer weather is better. A little bit of wind actually is better than no wind whatsoever. Now, a little bit specific on the larvae. How to find them? Answer, you need to open your eyes and look carefully. We all know what larvae look like, right? Well, some are hairy like this, some are not hairy. But key equipment could be things like a beating tray or a sweep net or learning to look for leaf rolls. So equipment might be your eyes or a camera. Leaf rolls like this. This is in a common umbellifer, not umbellifer, nettle family in Hong Kong. And larvae, they stitch with their silk the roll together at particular points, and the larvae live inside. Spiders do this too, by the way, so don't expect everything to be a moth or butterfly larva. Look for leaf mines, telltale grazing signs, either up the middle of the stem of the leaf, the main vein, or in the mesophyll layers. Each particular mine type is often characteristic of the species. And if you look at some of the UK websites, for example, they go into leaf mines and what they look like for each of the species. We haven't done that too much this part of the world. So there's an area, a niche, if you like, that is yet to be occupied. Some mines are very obvious. This one that looks like an exploding firework. Look underneath and you'll see a little case, and the larva lives inside the case, and then gradually at night, actually, this one eats away from inside the mine, and then by day back to the case. When the case drops off into the ground, then the larva will pupate, turn into the moth. Look also at plant galls in stems, in twigs, and buds of flowers. Fruit also, of course, have plenty moth larvae. You'll know about the codling moth that feeds in apples. You might know about the Mexican jumping bean moth that also feeds inside seeds. That's another both of those are tortricids. Rearing larvae takes a lot of effort. If you haven't the capacity, recommend you don't start without a little bit of background info and having lots and lots of boxes, particularly Tupperware boxes, ready and somewhere to store the food plant. Because these guys, you've heard of the very hungry caterpillar, they get hungry. So it needs lots of tender, loving care. And you need to make, because so many species haven't been reared through, making notes and take photos, take observations every which way. Now, that leads into photography. INAT relies on good photographs being supplied so that people who do identification have at least some chance of being able to identify what is offered as an observation. So what are you going to need? Well, a digital camera these days, because the days of celluloid and editing your photos before you take them are thankfully a thing of the past. You will need some kind of good capacity for data storage, because if you're taking lots of photos, you're generating lots of data. Your camera should be able to have some sort of good macro facility. So a macro lens, something like 90 millimeter, 100 millimeter, maybe 200 millimeter focal length. Camera phones also come equipped with macro options on many of the cameras these days. But what you're also going to need is the ability to close focus. So get very close to your subject so that you can see clearly features that give taxonomic information that enable a better chance of identifiers being able to do their job competently. Lighting for close focus is critical. Most examples in poorer light, and particularly at night, of course, you will need to be able to control the flash that takes or gives you a good light source for taking clear photos under poor lighting conditions. In really bright sunlight, also beware of high contrast that can wash out features that you need to see. The last point is what points of view, POV, to shoot that enable identifiers to see what's going on. So top down, dorsal view, sort of 3/4 view gives you good views, but also do not forget side on. Side on tells you so much about the head in particular, features like the eyes, the labial palps, the antennae. Also, you can see on this particular one, there are lots of tufts of scales on the wing. Those are often not visible from a top-down view. Also, if you're going to take a view from a species that rests in a tent position, tectiform, top down's not much help, doesn't give you much information. Look at it from side on. And also, if your light reflects certain scales, you might find a slight change in viewing angle will give you very different visual results. So these three pictures here are the same moth, taken from a few degrees difference only in angle. One, you have a big reflective patch above the two middle discal cell stigmata. This second one, the terminal area goes all shiny. The third one, all that shine, both medial and terminal, has gone. You can actually see the pattern on the wing. So providing multiple different perspectives, points of view is really important. Top down, side on, close on to the head are the three principal perspectives that I would request you try and take if time allows. If it doesn't, then try and capture something that gets a bit of everything. Jack of all trades photo sometimes is actually the best option. Right. Now you've taken your photo, how do you manage your data? Actually, what is data? For any biotic observation, the following is actually true. Once upon a time, we were confined to old school notebooks and celluloid, and of course, the old natural history collections. Digital these days is a much easier option for natural history, but it brings into play understanding database content, management, and how to use databases. Now, iNaturalist takes a lot of that problem away, but there's still a lot of information that an observation can provide if you make the effort. So the recorder, that's you, me, whoever gone out into the field, taken the photograph and provided the photograph for the observation. How did you make that observation? Was it an observation of something alive? Was it something that shows a trace of what that animal was? A scat, the pupil exuvia, frass, eatings, a construction, the case for the larva, perhaps. When? And this brings in another issue. 02, 05, 09 can be interpreted, depending where you are in the world, in three ways. 2nd of May, 02, 05, 2009. Oh, hang on. Is it 1909, 1809? So write the year in full. Write out the month, because it could be 02 as February, 05 as the 5th, and then 2009. That's how it's done in the Americas. Out here in East Asia, 2002 is the year first, and then the month, and then the day. So please write the month out. It could be abbreviated, but it's better in full. Write the year out in full. Now, identity. If you don't know what it is, don't try and assume that iNaturalist or any other citizen science database can identify it for you. AI is not 100% foolproof. In fact, it's a long way from it. iNaturalist AI looks at the external morphology, and moth taxonomy relies on internal morphology. So immediately there's an error in the system. Not AI's fault, not iNat's fault. It's just the way taxonomy for moths works. And AI on external stuff doesn't work the same way. So we must consider how to get around that. We'll come to that later. Now, if you didn't identify it and someone else did, the determiner, the identifier, put their name in, that's courtesy, And if possible, get the determiner to say where they made their identification from. What source? Was it a publication? Was it another website? God forbid it if it's Wikipedia, but it could be. What else? There's 5 points. Oh yeah, the place where you found this, or where you made this observation, and all that entails. Now, if you're going through voucher collections from 100 years ago, 200 years ago, the name of a place may well have changed in that time. So use latitude, longitude, please, or some sort of global referencing system for a grid reference. Universal Translocator is the one I use, UTM. But irrespective of that, INAT uses lat long, so go with what INAT uses, and the name of a place is not so relevant. If you have the time, record the habitat and possibly what we might call a microhabitat or the niche. If it's a larval observation, record the host plant. If it's a predation observation, what's the predator? I've mentioned weather already, but please do, in your own notes, record all the weather variables like the temperature, the average wind, how strong the wind is gusting. Is it raining? Is it sunny? What's the moon phase? That's important. Lunar activity determines how much you're going to see at a light source, for example. Full moon, not so good because it competes with your light sources. And then any other behaviors that you might have the opportunity to observe. Is the moth laying eggs? Is it nectaring? And if so, what's it nectaring on? Could it be pollinating? Are there pollen grains on the moth's horstellum, the tongue? Is it warming up, thermoregulation, or is it trying to cool down? Is it dipping its body in a stream? And how are you going to actually record all of that? Well, the old school way, notebook, photograph, written description, and of course, old school naturalists will have notebook after notebook. And this can be archived and is archived. And if it's looked after, it will last for centuries. Other ways of documenting include voucher specimens. And actually, this is critically important still, especially for tropical areas where we have not yet documented even half of the biodiversity of the Lepidoptera. So voucher material is vital for the description of new species. I'm not going to go there any further, just to say that it's necessary. Now, more modern stuff, catching up with new scenarios. I maintain a personal database on my computer using a relational database, Microsoft Access for one. And this can be customized for each user by each user. But one thing that comes across as an issue is when you have multiple different databases, how can you get those databases to talk to each other? If you're, how to call it, data gathering, data cropping, pooling data, the databases need to be able to be understood from one source to the overall repository. Online databases include iNaturalist, and that's probably one of the ways ahead. But there's a caveat. Like most things with moths, there's caveats. Back your own data up somehow on your own database and even print it out. Why? What happens if the internet comes crashing down? And it will one day, maybe. If we have a big solar flare that knocks out Wi-Fi, that knocks out power systems, we might simply lose all that data. So somewhere along the line, that data has to be backed up. Now, I've hinted at how you pull data together, and there is a modus operandi known as standards. And the key one for biological data is Darwin Core. It's not the only standard out there, but most biological data on databases that can be talking to each other uses Darwin Core compliant data. Now, what all that means is that each field in a database has a specific attribute, whether it's called for a species, a family, the recorder, the identifier, whether it's a voucher specimen, all of those particular components that make up an observation should be Darwin Core compliant. Now, on INAT, you don't have to worry about that. But if you want to keep your own database, please go ahead and search Darwin Core. Now, that's the end of what an observation is. How do you go about identifying? I'm going to talk about some key features that I would love to see as part of photographs made by observers. Visible field characters, and this is where we somewhat depart from strict taxonomic interpretation and start roughing it. Look for the wing shape, how the moth sits. Look, if you can, sometimes you can, at the posture and how the wings are coupled. That's getting a bit technical, might come back to that later, but coupling mechanism is one way of separating moths and butterflies. Look on the head, at the form of the antenna and of the labial palps. We'll come back to that in a moment. Does the moth have a horstellum? That's the technical name for the tongue, which generally is known as a proboscis for insects, but for Lepidoptera, it's one of the 37 taxonomic characters that separates Lepidoptera from everything else. Now, some moths, things like the Saturnidae, Atlas moth group, don't have a horstellum. Some species, families even, have scales at the base of the horstellum, the Gallicioidea. In fact, all galicids have scales at the base of their horstellum. Now, the pattern isn't restricted to the wings. Look at the head, look at the thorax and the abdomen, and not just on the top surface, the ventral surface. Look underneath the dorsal view. Legs also have specific morphology and patterns. Some of them got very, very kinky looking socks that are, or if you like, those who used to play rugby back in the 70s and 80s when you had stripy socks, striped socks. For larvae, side-on view, please, not just top-down view, because their morphology for legs, on the thorax, you have 3 pairs of legs, one each segment, and then down on the abdomen, you might have 4 pairs of legs, prolegs we call them because they're not true legs. There'll be the abdominal claspers on the back end. But some groups don't follow that pattern. They might just have 1 pair of abdominal prolegs, the lupomorphs, the geometridae, for example. And larvae have all sorts of pattern elements, lots of spines and other appendages too. Document everything, if possible, please. Right, wings, pattern elements. This is your basic grounding in your mapping, if you like, your mental annotation of what to look for and how to identify. So let me divide the forewing, in this case, the hind wing follows the same principle, into 3. There's the basal area, that's the bit next to the thorax. The medial, the middle bit. The terminal bit, that's the far end. Think of an end of a railway line as a terminus. So that's your basic ground plan for the basal bit, the middle bit, the far end bit. Add into that your edges and your corners. So at the base, well, that's the base, that's pretty obvious. But from the base to the top leading edge corner is the costa. From the costa down the outside of the wing is the termen, marries the terminal area. So termen and terminal, think far end, not too difficult. The junction of the termen and the trailing edge is called the tornus. Junction of the leading edge and the termen, the costa and the termen, is the apex. And the trailing edge is the dorsum. Now, on the hind wing, dorsum equates to the inner margin. And a lot of books use the phrase inner margin instead of dorsum. Now, in the following couple of slides, not all of the potential marks are illustrated. I'm just using some examples. So here we have vertical lines across the wing. From the base to the termen, basically you have a basal, a submedial, medial being the middle bit, remember? The medial, right in the middle, postmedial, post being beyond the middle. And then underneath or before, so sub, the termen, there is the subterminal fascia. So lines are all fascia. Add an E in, you get the plural, fasciae. Other options, you can often have a subbasal and a terminal. Now, what does that look like for real? We'll get there. Be patient. Wing patterns can represent like that. Can you see the subbasal? There's not a basal here. This is the base of the wing. That's the dorsum here. That's the costa. That's the termen. Can you see the subbasal? Submedial. The medial, the postmedial, and the subterminal. Yes or no? Can you see them? And if you can't, it's your lucky day because I've drawn them in. Okay, let me go back. Look again. In this case, the subbasal and the submedial have a dark area in between. So this could be called the subbasal submedial band. Look again. So it's the band is defined by those two fasciae. And typically the basal area, the medial area, and the terminal area are also split by the submedial and postmedial fasciae. So the divides into three and then subdivisions again with a fasciae. Right, moving on. Streaks. So going along the wing, parallel, roughly parallel to the costa and dorsum. You can have a streak anywhere on the wing. The four examples I'm showing are an apical streak at the apex, a terminal streak missing. This is the tornus. You remember, if I go back a couple on the wing area, the tornus is the angle between the dorsum and the termen. So this little streak down at the bottom corner would be a tornal streak. One at the base, basal streak. Two in the middle, the medial streak. Fairly obvious. Okay, now we're going to get a bit more complicated. Spots, aka a stigma or several or many stigmata. Spots generally are named after the shape they take. So 1, 2, and 3 are different shapes. One is roughly eye-shaped. Eye defined from orbis or orbit. Orbicular is the derivation. 2, roughly a club shape, claviform. The outer part of 4 is roughly kidney shaped. And if you know about your medical terminology, the kidney is derived from the renum, reniform being renal shaped, kidney shaped. Inside that, there's a spot in the discal cell. We'll come to the discal cell in a minute. But how do you actually equate what you see for real here to what you see in a museum specimen? After all, this is what you see in the field, right? Okay, so a little bit of jiggery pokery. Let's swap that around. This is just reversing the photograph upside down so that what you see here equates to what you see in a field guide, a book with voucher specimens. Let me just go back. Let's do that little trick again. Swap it around. And you can do this on your computer at home. Easy. Just use any half usable software app. I use View, which is very flexible, to rotate your image to what you see in the field guide. And it all becomes a little bit easier. So here is a small orbicular stigma, roughly on the medial fascia. Here you've got a, what's that? What's that? Is that a spot? Maybe it's inside an elongate spot, which I would probably call a reniform stigma. And inside on the discal cell, this is the discal cell here. Let me draw that in, use the laser pointer. So from here, going up, roughly like that, that's the discal cell. It's formed of two of the main stem cells of the wing with two crossbars. Sometimes there isn't a middle crossbar and it simply the two cells join so far, the two veins join so far down that you have an open cell rather than a closed cell with a crossbar vein. Let me get rid of those marks somehow. Start again. Flatten that. Come on, bye-bye. Get rid of that. Open that. And erase all of that. There we go. So we have four main cells. Watch what I say as a number 1, 2, 3, and 4. You can see what I call them on the sides here. On this example, the subcostal, you can see starts way down here. Let's try and draw straight. Not easy with a mouse. And it peters out somewhere up the top here. Underneath the subcostal is the radial sector stem, something like this, and it runs very close to the subcostal and then diverges. And then it splits into the radial sector, which splits itself. So that has 3 veins coming off, plus this one this way, roughly. The cubital vein stem is almost the same as the radial sector and then goes the other side of the discal cell. It then splits into the medial branches. So you have a branch coming out here, branch down here, branch down there, and a 4th branch there. Sometimes there are 5 branches, and usually all the branches split from beyond the discal cell. This is unusual. Finally, the anal vein way down the bottom there. Okay, so the 4 main stems, the radial then splits into the radial sector, the cubital splits into the medial vein, which should be veins here. These will be marked M1, M2, M3, M4, maybe M5. You can go look that up elsewhere. That's the Comstock-Needham system of veination. There are others that just simply number the veins, but it's easier to remember the names. Right, moving on. Patterns and colors. Well, patterns I've defined: the spots, the stripes, the streaks. But how they relate to each other, come on, click the right one, in terms of their color. Well, color is important, but remember, colors are mostly pigments, and pigments can fade under ultraviolet light, especially when the moth is resting up by day, for example. So colors are not necessarily a brilliant guide. Remember also that lighting, depending on your light source, the colors can vary. So it's helpful, but it's not necessarily diagnostic. And colors can change anyway, irrespective of whether the pigment's faded, because pattern elements can be different on a different form, or whether it's sexual form, whether it's a seasonal form, whether it's a form induced by temperature shock, or something to do with industrial melanism. There are lots of different forms. So there are plenty of spanners in the works when it comes to wanting to identify, because one species might have many different forms. Examples: these are all different plume moths. Wings of plume moths are difficult because they rub very quickly, they lose their scales, they lose their patterns. So look at other pattern elements elsewhere. And plume moth bodies, thorax and abdomen, tend to be spectacularly marked for each species. So much so that they are actually more reliable identification guides than the wing pattern. Remember also, underneath may be useful, in many cases is useful. Now, looking sideways, look at the antennae, and this is where you really appreciate having a good macro facility on your phone or on your macro lens for your digital SLR. Getting in to see how the base of the antenna is modified here. This is actually part of the antenna. That's the main part of the antenna there, but this is a modification of the base of the antenna. Likewise, here, that's the modification of the base. The main antenna here with the segments, the flagella, the rami, R-A-M-I, plural, ramus for singular, and then a simple thread-like extension. This species has a flattened antenna. This one, well, actually, that's part of the antenna too. Then the main stem of the antenna coming way out the side here. So look at, come on, the form, the length, and where the antenna are held. Is it a simple antenna? Is it got one set of rami? Does it have 2 or 3 or even 4 sets of rami? And is there some sort of modification at the base? All useful, but you only see these really with a close-up from the side. But they can tell you so much about what the ID of family and species. Now, I've mentioned labial palps before. What are they? They are a method of sensing carbon dioxide that's part of the mouth parts. Here, nice and easy to see. Normally 3 segments. I can do one bit there, one segment here, one segment in the middle, and then one segment at the end. And it's in the end segment, you can probably just about make out a little depression. Let's get rid of all of that. You can see somewhere down there, there's a kind of a pale zone. That is a pit or a groove known as Vollrath's organ that has little tiny, tiny hairs inside that sense carbon dioxide. Carbon dioxide is important when flowering plants produce nectar, high levels of nectar. They also produce high levels of carbon dioxide. And this is sensitive enough an organ to detect those high levels. And it's like us going down to the bar. Are there lots of drinks available? Yes. The bar's busy. If the bar's quiet, not many people there, not much left to drink. Same equivalent. And how do the labial palps vary? Answer is enormously, but you've got to get the side view. Absolutely fundamental. They can be forward pointing, they can be just tucked in under the head, they can be forward a bit and then up a bit and then curving round a bit, or a lot. Or they can be flat to the head in front, they can be projecting in front of the head quite a long way. One superfamily, the Gallicioidea, have their bottom segment forward, the middle segment up and slightly curved away from the face, and then the top recurved above the head. So all those that are segments round are Gallicioidea. You'll learn that that's one of the biggest superfamilies out. Learning how to differentiate them from the rest is very simple. But, Moths caveat as usual, look at this one here. It's recurved, but it's not recurved right over the head, and it's flat to the face, not far away from the face. Likewise here, slightly recurved, but flat to the face, not far away from the face. These 2 different superfamilies could be mistaken for Gallicioidea superficially on the labial palps, but other things tell you otherwise. And we can worry about posture and other components a bit later. Resting posture. I just mentioned posture, so here we go. How does the moth sit? How are the wings held? Are they tectiform, like a tent? Are they involute, like a rolled cigarette or a cigar? Does, or do, I should say it's in plural, do the antennae rest in a specific way? Are they short and above? You can't see the antennae there because they're actually tucked in under the wing. Antennae here are tucked in along the abdomen, also here along the abdomen. But posture also tells you, okay, this is a completely different group from this one. This sits up at an angle to the substrate with its back end on the substrate. This one sits with its head to the substrate, back end up here. This one sits with the wings flat in a wedge shape, also elevated at the back end. Plume moths tend to sit with the front end off the ground and the wings held out either in a T or sometimes forward in a Y. This one here is the same as this one here, family-wise, but a different subfamily. We'll get to that in a bit. Now, other things to note. What is the behavior? Can you document that? Well, yes, you can for certain things. Things like what's the nectaring source? And is it actually nectaring or is it pollinating? In this photo, you can't see pollen grains, even though there's pollen grains available. So this is definitely nectaring, not pollinating. Other kind of nutritional sources used, less savory, animal dung quite often, for example. Is there some sort of thermoregulation? Is it basking in the sun? Is it trying to warm up the wings by vibrating the wings? Is it exhibiting some sort of territorial behavior, fighting off other moths? Is there pheromone being sent out? And how do you know that? Is there observation of the pair? And what time of day is it all happening? Some of this will be nocturnal, some of it might be by day, diurnal. Each species will have its own time slot. Right, some resources. Now, when you go through the video, you can see this in your own time. I'm going to skip through these really quickly just to point out. Key books. This is one of them. It's my go-to for a lot of the functional stuff. This one is key for all the natural history stuff I've talked about. This one, if you can get hold of it, is basically the Bible for the technical stuff. And it is technical and it's a heavyweight book. But there we go. This one is the most recent one that documents families for India, not just for India. It actually lists all the families. So it's relatively up to date, published just last year. And this one gives you an up-to-date rundown of the biology and of evolutionary history. I have no commercial interest in these books, by the way, even though I'm a co-author on one of them. But in terms of their validity, these should be on your library shelves, especially this last one, if you're interested in finding out about how to understand more about moths. Butterflies kind of as well, but it holds true for them, but moths in particular. Now, some web resources. Don't try and note this lot down. It'll be up on the video later. I'm also going to put this slide, in fact, the whole presentation up onto slide share. So you'll find that these names, resources, are links. They are linked to various websites and journal websites. So you can go and delve into this lot in your own time and learn from them. These are not linked except the last two key references. These are more key references, and some of those I have linked to. And there are even more. This is macro moths now, not just micros. All important. All will give you a sense of understanding of relationships between species and families. I'm going to hold it there, put this onto pause. Where's the pause button? And we will stop the slide share for a bit. Okay, back for my screen. Ashwin, are you still with us?
Yes, definitely enjoying the session.
Okay, I'm going to open this up for the next, ooh, where are we at? 5 minutes for questions. Anybody who's got questions on how to make an observation and what to do to look for identification features.
Okay, so we got a couple of questions on the chats. I mean, actually, quite some numbers. I'm glad that people are enjoying and having a lot of questions. So one of the question was you demonstrated or you showed various ways by which moths can be brought in for observation. So there was a question
Mm-hmm.
that is this method safe for rare moths and how do we sedate them?
Um, I'm going to go one back from the rarity issue. Forget whether it's rare or common. Actually, they're both important. Common ones make up the bulk of biodiversity. So don't hang up yourself stuck up with is it rare or common? It doesn't matter. What you observe matters. So it doesn't matter what POV you shoot, as long as it's something that helps you identify what you're looking at. Really, don't get hung up on rarity. It's not important in terms of making an observation.
Yeah, right. Okay, so the next question was, what are some easy or alternative way to build own cheap or easy DIY moth trap or an insect trap, especially for students who can't afford, to make a elaborate one. Any
I would
tips on that?
suggest you go out on the internet and search. There are lots of how-tos. National Moth Week website has lots of video resources on how to go on and do that. If you go on to the mothing and moth watching Facebook forum, there are chats. Search on those. You'll find it. No problem. Chitra, you raised your hand.
Roger, what a wonderful presentation. I hope we can listen to you more presentations. So my question is, the very less studied caterpillars of moths, so do we have
Yeah,
any
off my radar a
catalog?
bit.
Do we have a catalog for Asian lepidoptera caterpillars? Because it's really
No,
very difficult.
no, it's a big area that everybody can really help contribute to.
Yes,
Um,
yes,
I
yeah.
won't say it's off my radar because I've read about 100 species, but I mean, 100 out of how many thousands? It's a drop in the ocean. If we get, let's say, 1,000 people doing Asian H Challenge, putting in a life history of a caterpillar, um, that's 1,000 more species perhaps looked at. It's not difficult to rear one or two. It does take a lot of effort,
Yeah.
but if we can all contribute, that enormous wall of unknown becomes quite a lot less. So, resources for caterpillars is really what you should be asking, I guess. Yes,
Yeah.
there are some, but not many on INAT. Um, there's a good website on UK moths that does caterpillar illustrations. Check it out. Not UK moths. There is actually a Lepidoptera larva thing, and there are several Facebook groups dedicated to caterpillars of Lepidoptera. What I will say is that when you look at a caterpillar, look at the number of prolegs on the abdomen, because that is really important. One proleg pair will be geometridae, two pairs will be erebidae, three pairs will be erebidae or noctuidae, four pairs is pretty much everything else except the zygonidae and the lemacodidae, because they're different. I'll come on to that after we chat and we get back to the particular families. Okay?
No, but see, like for example, we find some caterpillar in the forest. Like I
Mm-hmm.
recently saw Urina caterpillar. Means I think it's Urina because it's called Mad Hatter and it's got a very specific head stacking technique. But we
Right.
have an adult reporter from India, Urina lobega, but I am not seeing. That is a gum skeletonizer, and I'm not seeing that host anywhere close by. So I'm more
Well,
interested.
the only answer really is to get out into the field and do the legwork, not for the caterpillar, but
I'm not able
rear
to
the
bear that
caterpillar
because it's a
and
forest.
associate the host plant. Make sure that you see the caterpillar eating the host plant before you take it away and take some of the host plant. then rear it through under lab conditions. You will need a Tupperware box and lots of cleaning. Every day you've got to clean the thing out, make sure it doesn't get too humid, make sure it doesn't get too dry. It likes the Goldilocks zone.
Yeah, I did rear Camellia, but I was not successful in rearing this because
it's fungi are a problem, bacteria are a problem. You really do need to be ultra clean. Things like Dettol to wash out your rearing box every time. So cleanliness is next to godliness is the phrase, and for moths, it absolutely. So lots of care needed, and lots of willpower. Just get in there and do it. I'm going to have to cut time there for questions because we've got another
thank you
half an hour to go. So everybody has taken a quick drinks break, I hope. I am going to put myself out of view and I'm going to go back to the screen share. Right, find the screen share. Where are we? That one. Come on, screen share is, get that out of the way. There. Right, screen share is on. So this is a rundown, more or less family by family in approximate taxonomic order, but I'll be dealing with micromoths first, and then with what I call Mesolepidoptera, which is technically micros, but they look bigger than most of the micros. And then I'll wrap up with macros, and I've got 20 minutes. Good luck. Right, I'm going to go through this fairly quickly. I will not read off what's on the text for each of these species. You can do that. I will read out a few notes I've made that complement what's on the screen. So first up, I'm going to get rid of the, where is it? Muffin. That one. No, not annotations. I'm going to have to live with the, where is it? Pens. Pointer options, that one. Yeah. Right, that's better. So, clothes moths. They all have some sort of case, some of which you will know because they do recycle your furs. Bear in mind, these guys have been around millions of years. We've only been around a few 100,000 years. We're the ones who are the pests. They are designed to be sustainable users of resources. Homo sapiens has got a lot to learn. Now, I've said Psychidae, aka bagworms and sack draggers, but we'll get there in a minute. What I want to show for the adults is basically they almost all, one exception that's here, have punk hairdos. You go back to the 1970s and you saw the spiky hairdos in London. These may not have pink tints to them, but some have got orange, white. All of these have very spiky groups of hairs, cilia, the scales, modified scales on their head. And it is not completely diagnostic, but it points you towards the clothes moth. And it's the first port of call. If you see spiky hairdo, you think Tineidae first. Close relatives of the bagworms, the sack draggers, they make bigger cases on the whole. Some of these can get to nearly, 5 or 6 centimeters long. They're an incredibly important group because they have a number of species that reproduce parthenogenically. And they make superb ecological studies. Won't go into it now because we don't have the time. But do look for the discal cell being heart-shaped. That is found, I think, in no other group. Not all psychids do it, but some others have, in the subfamily Orchetinae in particular, a second cell mark that's actually a split of the anal vein. Move on. Right, the next major group of micros, the gracillarids. There's actually a superfamily Gracillarioidea that contains 3 families, 2 very small families. All are in the auricular region, but the main family is the gracillarids, and they are leaf and blotch miners. Some will form just complete blotch mines, some just make a gallery mine, some do a bit of both. Some curl leaves at the edge in early instars and then form a blotch mine later on. The form of the mine is often very distinct and can actually tell you which species you're looking at. Here, this is Phylacnistis citrella. Those of you that try to grow your lemons and oranges will know this for sure. Tiny, tiny orange-yellow caterpillar leaves a rather a nasty trail of its frass inside the gallery mine that eventually kills the leaves, because if you get too many of these in all the leaves, the plant dies. Not so good. Over 2,000 species globally, and adults, well, they can look roughly like this. 5 major subfamilies within the gracilariids. Resting posture is usually upright off the front legs with the back end on the ground, but can be like a bridge almost, with the whole body off the ground or the front of the head just slightly off the ground. Usually very ornate markings, but can be quite simple. These are tiny guys, by the way. We're talking from the edge of the labial palps to the end of the termen is typically less than 5 mm, not big. One of the field characters is that the antennae, which are very long, often get waved around like somebody doing semaphore signaling from the Navy before the moth settles down to rest. Next lot, Eponomutoidea, okay, superfamily, includes a number of families that were once upon a time all regarded as Eponomutidae. Hang on a minute, plug battery in, that's done. Close that, there we go, battery's up and running. Sorry guys, girls. Okay, these guys, many larvae are communal web spinners. It's really important. So you'll see a silken mass with tiny, tiny larvae in it, and it may very likely be one of this group. The posture, unlike the gracilariids, this is typically head down, butt in the air, rather than head up and butt on the substrate. Not all are like that. One of the main groups, the Plutelidae, this thing here, which most people will know as the diamondback moth, as a major pest of Brassicaceae, sits flat to the substrate. Long, very what we call porrect labial palps. Some species will labial palps that are up curved, but don't curve up above the head. Next group, the Coryutidae. These guys are mostly diurnal, active by day. Again, small. Most coryutids have a wing span, i.e. length from base to apex, of 5 or 6 mm only. Some a bit bigger, most not. And they jump and hop and skip around, especially on figs, ficus. Not all are on ficus, particularly in the temperate zone they use other resources, but in the tropics it seems that figs are the main larval host. The Brenthianae, one particular subfamily that have lots and lots of eye spots on the terminal area, are supposed or thought to mimic jumping spiders, the Salticidae. And when you see these guys on a leaf jumping around, you can see why. And it's thought that the jumping spiders actually don't predate these guys because the mimicry is so effective. They avoid predating the moths. The spiders avoid predating the moths. Very distinctive pupa inside a spun. It's not a cocoon, really, is it? It's a shelter. It looks a bit like one of these sort of fancy modern tents that's pegged down in a few places. Rather nice. Right, plume moths. Strictly speaking, this is out of taxonomic order, but in terms of coverage, you're getting them now, not later. Very distinct group, resting in a T or sometimes a Y posture, with the forewing split into 2, the hind wing split into 3. But when you see them at rest, often they're rolled up so tight you cannot see the splits, more like this one here, or perhaps this one here. The split, the cleft, if you like, doesn't always go right the way down the wing, might only be in like the terminal zone. So actually trying to ID these guys from the wing patterns is very difficult because the wing is rolled up, so you don't have a pattern to see. So look at the body, look at the pattern on the body, and they are different between every single species. Right, now the next half a dozen or so families are all back to Gallichioidea. By now, you should recognize palps that come out a little bit, round a lot, and keep going round and over the back of the head. That is a Gallichioidea thing. Not 100%, but 99%. So there's always a few rule breakers, as I said, in the moth. Moths, they're a fairly rebellious lot. Okay, Autostichidae. Males and females often have different labial palps. Two segmented in males, three segmented in females. But the male two segments, this is a female, the males have the second segment going all the way around and over the head, thickened all the way. I've marked that up here for the periacmene. Hind wings are generally fairly full. These are representative taxa, generally oranges, blacks, yellows, and whites. Generally, there are plenty of exceptions, a pair of discal spots and a second spot separate. So you get kind of like double spot, single spot, double spot, single spot, double spot, single spot. You get the idea. Double spot, single spot. Not all, but most. And then a posture that is not tectiform, not tent-like, not involute, somewhere in between, slightly rolled. Next up, close relations, the Echophoridae. These are generally a bit smaller, always three segmented on labial palps, always back over the head. Generally, with a more tightly rolled wing posture, but the fringe on the termen very strong. Not always, but generally. These guys are tiny. The forewing from the head to the apex, typically 3 to 4 mm, sometimes a bit bigger, but not often. Unless you're in Australia, in which case, a whole nother ballpark. Okay, the next Gallichioid subfamily I'm going to deal with is the Cosmopterigidae. These have very fine, slender, scythe-like palps. Again, thin-winged, like the Echophoridae, and usually with a big fringe at the back. Sometimes the legs are modified with big spikes or whorls of bristles. We'll come to that later because that's significant. It's another family. Some more examples. Behaviorally, some of these are a bit like whirligig beetles, but on a leaf. They dance in a little pirouettes and all sorts before they settle down. Combine that with the red eyes and the very fine scythe-like labial palps, and you can be pretty sure it's Cosmopterigidae. This guy excluded. The family Scaezo, I can never say it, Scaeozophinae, much flatter. They don't have the obvious palps, scythe-like, and generally much blander looking things, and bigger. Cosmops, these guys are small, and the smallest 2 millimeter wingspan. Tiny, tiny, tiny. A bit bigger, the true Gallichidae. The hind wing tapers at a point to the apex in sometimes a very obvious finger-like protrusion, but always projected. Forewings usually full, hind wings usually full. And the palps, typically with bushy second and first segment, and then a peg-like, sometimes curved, final third segment. Some examples. We get there. Here we go. So these are Dichomeridae, peg-like, fine palps in bushy bits, all the way through. That's really Dichomeris orientis, very, very obvious example. And then the Thiotrichinae, thinner wings, very fine hind wings, and cosmopterigidae-like palps, although the second segment usually a bit bushier, just like the Dichomerididae. Eyes, black, not red, like cosmopterigids. Okay, more Galicids. This is Moth subfamilies, and you'll see, again, bushy second segment labial palps, recurved, strong third palps. This guy's a bit different. The Anacampsinae have very heavily scaled second palps and slightly weird upright and backwards labial palps for the final segment. These are generally blander. There we go. You will have noticed from the previous lot, those guys are colourful, so don't think moths are boring and brown all the time. They aren't. Now, I mentioned earlier about whorls and bristles on the legs. This is more usually associated with the Stathmopodidae. This is another highly diverse group in the Orient region. We're looking now well in excess of 600 species. Even in Hong Kong, we're at about 40 species, most of which are yet to be described. But they almost all have these kind of cow-horn labial palps when you look at it from above. From below or to the side, you can see the palps are actually very, very much tucked in under the head and recurved, not quite over the head like most Gallicioidea. Some don't have quite such obvious cow horn to the side, but protrude more to the front. The other main factor is the whorls of bristles on the hind leg, and typically raised hind leg resting posture, just like this one. Sorry about the focus on the head. I was focusing on the whorls. Here you see again the Stathmop with the cow horn palps, raised hind leg, and whorls on the palps. Some of these are incredibly brightly colored diurnal species. This is an extreme example. You have the cow horn palps, the whorled bristle legs, and the long slender wings. Here, just to prove there's an exception to the rule, this one's got modified base of the antenna. The base of the palps are to the side, but the palps don't do the cow horn thing. They just come up and back over the head. It is bristled on the legs, but not in whorls, and the leg isn't held upright and out. So be warned, there are always caveats and exceptions. Next group, the Xylarictidae. These are bigger Gallicioid micros, much stouter. I've given you one of the main characters there. Now, just a word on technical terminology. Tergite, you can't see because it's part of the body. Likewise, the sternite. The body is divided into the dorsal and ventral exoskeleton, and each segment, you know, insects are segmented animals. Each segment is divided into the tergite for the dorsal part to the sternite for the lower part. There you go, learn something new every day. But Xylarictids generally tectiform, tent-like posture, relatively stout and chunky and large, and some extreme. This one palps like no other. Quite a few have these ridges. It's a group that in Australia goes absolutely bananas. I'm not going to go there, but it's just an amazingly diverse group, and many species in Australia are awaiting identification and formal description. Right, the next, and I think this is the last group of Gallicioids I'll deal with, is the Lepidopteridae. Get your teeth out of that one. Typically really long antennae pointing forwards. Again, the slender scythe-like antennae, typically not red-eyed. Quite large, broad wings, both forewings and hind wings. Posture is important here. The Taurodorini, which is one of the two big groups within Lepidopteridae, have this wedge posture with the antennae out forwards. Lepidopteridae, the nominate subfamily, tend to be thinner winged, not always with the antennae to the side, quite often to the fore, like this one, and the posture with the body flat or slightly raised at the front to the substrate. Another typical example, this is a Lecithocera species, typical dull brown. Unlike the Orthostichidae, a single spot, 4 in the middle, and then another single spot with the dorsal, joint smudge, if you like, towards the torus. So not double spot, but single spot, and the antennae pointing forwards. Caveat time, isn't there always? Okay, this is a Lecithocerini, but the genus Nosphistica, and they are just weird. End of. Just watch out. You'll find them, but not many. Back to Toradorini. So again, this is the fancy pants job with this dual color. Every segment alternate color. Antennae, but antennae forwards. Scythe-like palps. The back end up in the air. Typical markings. These are generally a bit better marked in the Toradorini than in Lecithocerini. Okay, let's last lot of micros we're dealing with is another big group, the Tortricity or bell moths. 3 main subfamilies. This is Clidonotini represented here. Small group, but they're widespread throughout the Orient. Tortricini, much bigger group, and a lot of pest species in this, the fruit and leaf rollers in particular. The posture here tends to be a very flat involute, so gently rolled, or sometimes a little bit sort of flat and angular, wedge-shaped. Pattern elements here tend to be fairly mundane browns, sometimes whites and yellows. Not easy to identify. Nor are the Olithrutini, which is the biggest subfamily, divided into half a dozen or so tribes. This is a group that are generally fruit predators as larvae and often very colourful. One characteristic, not unique to the Tortricity, but very distinct for them, are these little markings on the costa, which we term striguli. Most Olithrutini have them. Combine that with the short antennae and the chunky posture, usually tectiform, sometimes a bit involute, and often, it's not easy to see here, spoke-looking eyes. So if you think of a car that has 5 radial spokes from the center to the tire, this is almost the same. And move on a wee bit. Can I see this in any of these? Yeah, this one here, small center and then little radial spokes coming out. That's a classic Tortricid thing. But if you look at the posture, generally tectiform or slightly involute, but antennae short, held slightly upright, and quite colourful on the whole. Right, Mesolepidoptera. Knot horns, meal moths. This is Pyralinae of the Pyralidae. Note the flat posture, chunky. Abdomen has the antennae absolutely tight to it, slightly splayed apart, but not often, sometimes right down the middle. The Pyralidae is an enormous group, 9,000 species, maybe more, of which 2,000, 3,000 in the Orient, maybe more. Not well documented yet. It's a major group for what we would term pest species. Let me put it another way. They are excellent recyclers of detritus. That's what they are evolved to do. And detritus, as far as they're concerned, is the stuff we drop on the kitchen floor. It's the stuff that we store in our wardrobes. They regard that as material for recycling, doing their job. Other subfamilies, the Galeriines, are typically smaller, very involute to slightly flattened, and often associated with Hymenoptera inside beehives, inside wasps' nests, as larvae, not as the adults. Okay, the Epipassionae is a weird, wonderful, and wacky group of moths that is primarily oriental tropical, heavily modified, both labial palps and antennae. The larvae are usually gregarious, forming silken, rather mucky, frass-laden webs inside lots of rolled leaves or stitched together leaves, and the wings usually with some kind of modification of scale, presumably as scent glands. Pyralinae are the other chunky group in Pyralidae, usually slightly deltoid shaped, resting typically fairly flat, maybe slightly tectiform, maybe slightly involute. Antennae almost always over the back of the abdomen, slightly splayed. The next group is taxonomically very challenging indeed. The Phycitinae, they are another big group of plant predators and aphid predators. They do an excellent job on controlling a number of aphid pest species. Elongate posture, antennae modified at the base for every male species. If you zoom in on these, where's my zoom in? I know there's a zoom in here somewhere. Come on, that one. So zoom in, you can see the basal modification to the antenna. That is a male thing in all Phycitinae. Female phycets, this one here, for example, don't have the antenna modified at the base. All are elongate forewing, fairly broad hindwing if you look at voucher material, but the hindwing is tucked away nicely, folded away, and very elongate posture, but usually resting either flat to the substrate or slightly protruding from the substrate at the front end. Right. The other Pyraloidea family is the Crambidae, and I'm going to rattle through these really fast indeed. Many subfamilies, each of which is quite characteristic, but again, the key thing that tells you it's Crambidae rather than Pyralidae is the posture and the general structure, shape, whether it's, what am I after here? Stout-bodied or slender-bodied. Most of the Crambidae are slenderer than the Pyralidae. All have their antennae pretty much back over the abdomen or slightly splayed. You'll see that throughout the photos to follow. So Crambidae, usually grass associated or open habitat associated, sometimes wetland associated. The Axentropinae or Nymphilinae of old are associated with freshwater, sometimes intertidal water, whether that be brackish, whether that be still water, or whether it be running water, take your pick. There are species for every niche. The larvae are aquatic. Spilomelinae. This is a huge subfamily, generally with a deltoid posture, sometimes a bit more splayed, flattened out, can be very flattened in some species. Antennae always down the back or raised over the back a little bit. Many pest species in this group, or at least competitors for our fruits and vegetables. Some are very colourful, some not so. Some diurnal. This one's a diurnal species here. This can be diurnal. Canagethys can be diurnal. Spaladia can be put up in the thousands at the right time, right place, by day as well as by night. The Pyraustinae used to be grouped together with the Spilomelinae as a smaller group, generally slenderer, more elongate, often long-legged, often associated with herbs rather than woody plants. Mint is a particular known group for larval host of this lot. Right, Thyrididae, tropical leaf moths. Not going to say too much on them other than they're colourful, they're wacky, they have very distinctive posture, and they're an absolute swine to ID because a lot of them look pretty much identical. The genus Herdonia is a classic example. Inoue treated this in 2 major papers. I'm not going to say more other than that they're tricky. Good photos required. Colourful group. Right, lemicodids, not going to say too much. They are generally small, they're known as cut moths or slug caterpillars. A difficult group to ID again because lots of similar adults. Whole groups that some of these have been revised fairly recently. Peraza now completely reevaluated for our part of the world, but not in the neotropics yet. The larvae have retractile heads. This is a Zygenoidea thing. And in Southeast Asia in particular, they're associated with palm, but they are either stinging type or slug type. Mark Epstein and company have just reviewed the whole systematics of Lemicodidae. Go look that up. I'm not going to go there, but they are another weird and wacky group, and you'll see them by day. Okay, I've mentioned Zygenoidea just now, and retractile heads. You look at this guy here. Let me go back to that zoom element. Come on, pick that up. Thank you. And you can see the head here partially covered by the first thoracic segment. This is actually a flap that the whole head can tuck itself in under as a protective mechanism, as if these guys need protecting. Zygenoidea are colourful for a reason. This group make hydrogen cyanide as a protection. Hydrogen cyanide, those of you that follow history will know that it was the gas used for exterminating and genocide in the Second World War. Not a good thing to do. Right, moving swiftly on. Adult burnet moths are also colourful, again, because they have kept the hydrogen cyanide and can still emit hydrogen cyanide. And a lot look like some of the day-flying moths from the tiger moth family, the Arctinae. More on that in a bit. Oh yeah, other day-flying. We mentioned pheromones earlier. This is one of the big groups that we use pheromones for. They do sometimes come to light, but if you have pheromone traps, this is the sort of thing you're looking for with pheromone traps. In Vietnam, 30 years ago, we knew 3 species of Cecidae. Now we know 300. Why? Because we have Cecid traps with pheromones being used to record moths from this family rather than casual observations from before. Big shout out to Sasha Haldane for this lovely photo of a clear wing moth, aka Biminic Cecidae, seen in Hong Kong earlier this year. They are a fabulous group of mimics, one of my faves. Now, more Mesolepidoptera. This is the Coccidae, and they are actually quite big, chunky moths on the whole, not to be confused with true macros phylogenetically, but they are quite primitive. In terms of their visuals, though, big moths reticulate complicated patterns, very short section of heavily pectinate antennae. Let me zoom in, come on. Very short, maybe 1/3 at most, is bipectinate and heavily so, and then the rest of the antenna simple. A few more examples, and then we move quickly on. Again, the short pectinate, the rest, often with the abdomen protruding, and a lot of these are associated with wood boring or stem boring in either trees or in the Poaceae, reeds, bulrushes, and so forth. Big chunky guy. This one doesn't have the pectinations at the base of the antenna, but it does have the rest of the typical broad wing reticulate patterning. Another one, this incidentally is egg mass at the back end. Chunky body, big reticulate pattern element. This is a very big moth, big as my middle finger. Now the Metabelidae are technically super set out from the Cosidae nowadays, but within the same superfamily. Also, less patterned, generally simpler patterns, pale browns on cream with dark brown markings and protruding body for the male. Right, true macros, Drepanidae, hook tips, caveat. This one is a Drepanidae, but it looks like a Geometridae, so do be warned. Visually, AI would put that as Geometridae, but it's not, it's a Drep. And that's because we know the tympanum, the ear, is on the second, not the first, abdominal segment. So that's how you tell Dreps from Geos. The Geos also have an abdominal ear, but it's on the first segment. Now, then other examples. Hook tips, well, because the tip, the apex of the forewing is hooked. This is a Tridrepana species, quite classic. Some are more extreme. This is an Areta species. Some don't look like hook tips at all. They look like noctuids. So there used to be the family Thyrididae. Now the subfamily Thyridinae is phylogenetically part of the Drepanidae. This looks more cossid-like than or noctuid-like than a hook tip. But there we go. Just had to live with it and learn all the bad boys who break the general rule. Some Dreps, well, yeah, go figure. Very weird indeed. But that's a Drepanidae. So the breadth of variation amongst posture is enormous in this group. There you go. That's more classical Drepanidae. And again, okay, Notodontidae, so-called prominent moths, because they have prominent tufts of scales, mostly, not always. They are forest species by the main part. The larvae are on forest trees, and enormous diversity in truly unmodified forests, but very, very much scarcer in modified habitats. Now, Erebidae, this was once upon a time Noctuidae. Within Noctuidea, quick explanation. Counter tympanal hood. That's a little cover, a bit like our earlobe, on their abdominal segment, and it is a projection that covers the tympanum, the ear, which is on the third thoracic segment. It is a feature that defines, in part, the Erebidae. But in terms of how you recognize them in the field, you have to live and learn. You observe, and the larvae are possibly easier, simply because they have 3 or 2 pairs of abdominal prolegs. This is a Hypena species. 1, 2, 3. That's the real legs. That's the abdominal prolegs, and that's the anal proleg, anal clasper. So when you count proleg pairs, these are the ones where you should be looking. This one doesn't have a 4th here, which is the basic ground plan for Lepidoptera. Excuse me. Eye spots are a fairly common theme on Erebidae, as is UV reflectance. And this one you can see UV reflectance on the terminal fringe and on the body. Also, patterns tend to be cryptic for the forewing and quite often what we call fright. What's the word I'm after here? inducing eye spots and the like on the hind wing. So the crypsis is primary method of defense. If that gets busted by the predator, the forewing is lifted, revealing bright colors for the hind wing. That makes the predator, whoa, what's going on? And gives the moth time to get away. Right, some other examples included in Erebidi now are the tussock moths, are the tiger moths, of which there are many. And then to noctuidae, the countertympanal hood is slightly different. The hindwing venation differs from that of all Erebidi. You can look that up in the Riza Zahiri papers. But in terms of diversity and morphology, generally they are tent-like, they're tectiform, and with chunky four-paired proleg larvae. Cutworms is the phrase that comes to mind, and they are primarily monocot feeders or herbaceous feeders. They're also exceedingly diverse in cooler, temperate or montane tropical areas. And amongst many species, there are notable migrants. Bogong moth, Peridroma salcia, what we would call in the UK the pearly underwing. One of the world's most widely distributed species, Agrotis ypsilon, aka black cutworm or dark sawgrass, are all noctuidae. Now, the nolids is the last of the noctuidea I'll deal with, and invoke John Horseman's rule. If you can't identify it as a noctuid or an erebid within the noctuidea or even some of the other families, it's going to be a nolidae. Now, here, the key thing for the definition is actually the boat-shaped cocoon. Not going to go into that anymore, but that's what a cocoon looks like in the nolidae. As to their adult appearance, typically tectiform in posture, but not always. Some are quite small, like Nola and Arius. Some get a bit bigger, Blenina, and other genera within the Carainae. Now, that's enough said about them, enough time we have. Uranidae, sunset moths, scoop wing moths, go from the very big to the not so very big, but are primarily broad-winged or scoop-winged. You can see for the hind wings here, and it's clear on the next view, the incredible modification of the hind wing shape and also to some extent of the forewing shape, often with little tails. True geometrically usually don't have tails, but there are some exceptions. Rounded wings and very flat, broad-winged posture. Multiple subfamilies, but primarily feeding on herbs in the Laurentiines and the Sterines. The Geometrinae are on shrubs generally, and the Inomini are primarily forest species. But what ties them all together, just one pair of abdominal prolegs. Let me zoom in. Come on, zoom. So you see one pair of abdominal prolegs, the anal claspers, and then nothing all the way along the abdomen until you reach the thoracic true legs. Right, last major group, the Bombicoidea, include the infamous, collectible, unfortunately, Saturnidae, so emperor moths, atlas moths. These are two from Hong Kong, but they can't feed as adults. They do all the feeding as the larvae. They are amongst the biggest oriental moths, not the biggest in the wingspan globally, but by wingspan and wing area in oriental, certainly. And they have very chunky larvae as well that conform to the 4 abdominal prolegs and anal clasper format. And I'm going to finish with the hawk moths, aka sphinx moths if you're in the Americas. Deltoid wing shapes typically. Larvae are classic 4 pairs of abdominal prolegs, but with the bombicoidea caudal horn. So on the anal butt end, they have a modification that is basically a diversion technique to take predators away from the head. You will probably know about this guy, squeak moth, aka Silence of the Lambs, death's head hawk moth, bee robber, so on and so forth. The oleander hawk moth, armyworm moth, one of the classic species in the Oriental region. Big, chunky, very strong flight, and strong nectaring. They are a key pollinator. Now, for Asia, wrapping up quickly, websites, Moths of Borneo, one of your major go-to resources for identification. Looks a bit like that. No, now we have volume 13 online, not listed in this group, but unfortunately, a lot of the links in volume 13 are not valid links, and you don't get to see quite a large proportion of volume 13. Most unhelpful. There's another part for Borneo covering the pyralidae and thyrrididae. Actually, it's not just Borneo. The thyrrididae in particular are expanded by Terry Whitaker to include much of Southeast Asia and South China, so a very useful resource. The Moths of Japan website is as complete a website for a country as we have in this part of the world. Don't be put off by saying Japanese text only. It lists all the families in the scientific names and has very good links to pretty much all the species that are covered by Japan. And if you use a online translator, then you can read the Japanese without too much hassle. Comprehensive and pretty accurate. Moths of India, new kid on the block. It's only been around a couple of years. It says since 2015. It doesn't feel that long, but the coverage is getting better. Accuracy is getting better, but still I have to let Sanjay know of a few errors every once in a while, but on the whole, out of the thousands of images there, pretty good job. Now, last, I think, of the websites we're going to mention is the Philippines Lepidoptera, which is slightly differently arranged by album rather than specifically by family, but again, quite a lot of photos, much less text than, say, for example, the Moths of Japan or the Moths of Borneo, but nonetheless a good regional resource. And of course, iNaturalist has projects for all Asian countries. So if you need to go and find something from your particular region, don't hesitate to look at the project portfolios and see what's there. Okay, on iNat, so here we have examples. We have Moths of Asia, but what do we do to actually put our data there? Well, good photos. If you're uploading lots of photos, please try and down sample your photo to 1024 pixels width and crop so that the photo features the moth rather than having a tiny speck in the middle or off to the side. Naming the file is really helpful. If you can, and you know what the ID is, include the file name with the species binomial. So here, CRA is for cranberry. PYR is for pyrostomy, the subfamily. So you've got family, subfamily, genus, and species. iNat can strip out the binomial from the file name automatically. So when they get uploaded, I don't have to fill that in. It's done for me. When you're putting in lots of observations, don't use the app. Please use the website because it's so much easier if you have access to a laptop or a full Mac computer. Otherwise, it's a grind. Mark in observation data so you can batch upload. And then when you're uploading, you have, come here, the option to add in information, not just for the identification, but what you've seen in terms of an adult, larva, was it dead, was it alive? All of that is really useful. But in the process of IDing, don't rely on what INAT tells you or what Wikipedia tells you. Please check it out for yourself. If you know what family the moth comes from, and I've given you a rundown of the common families in Asia, put the family name in, if you're sure. If you're not sure, just put Lepidoptera. That'll be a start point. Please also remember that not every single moth species is identifiable from photos. Actually, a lot aren't. There are many, many cryptic groups, and there's no way I or anyone else can tell what the species is from the external habitus. So if you put it in as an ID and you're wanting to check, use the compare function to see what is around you locally, not using the ID suggestion, but the compare function, because then it gives you similar species in the group, and you can drop down from the species to the tribe to the subfamily and down the hierarchy to superfamily. And you can narrow or broaden the location as you like. So it could be Hong Kong, it could be China, it could be Asia. Or you can just drop the whole thing to see all the species in a particular genus or tribe from the globe. And then you see what it looks like, what are the main photos, and where it's from. And you can compare. Does it look the same? Yes. Then you select it. Does it not look the same? Does it look similar to something else that's suggested? Select that instead. Once you've done the ID, please annotate at least whether it's dead or alive, and at least whether it's an adult, whether it's the pupa, whether it's the larva, or whether it's the egg. These are really, really useful for analysis of data, whether it's dead or alive, tells you which stage and which time of year. Very, very helpful. Now on INAT, what else can you add that's really useful? Go back to my slide, what's an observation? And you recall that actually the time, the place, and the recorder and the ID is only 4 of 9 or 10 particular points. And I kind of skipped over a lot of the ecological data. But INAT offers the option to add in tags that you can type in as a freeform tag for National Moth Week or for Asia Nature Challenge 2025. Whatever you like, you can tag it with that. The observational data as attributes from the annotations is also included in observation fields, and then you can actually add in external information, whether it's ecological or other matter, doesn't matter, but extra information that is related to the observation goes into the observation fields. You can choose from several thousand fields. What is a field? Well, an observation field is a database data field defined by a name and then described. And you can actually, if you can't find a field that you want to add, you can actually add these yourselves. So every day people are adding new fields, and that is both good and bad, because actually overall there's a lot of replication of observation fields. But if you want to find out how to add, go look, go find, go play with INAT's observation fields. Important ones will be things like abundance, who's done the observation, who's the determiner, i.e. who's identified it, and you can add that information very easily. Now, I'm going to round up quickly on INAT, some of the projects. Moths of Asia is an umbrella project. It pulls in data from other collection and original projects, such as Hong Kong Moths Project, Moths of Taiwan, Moths of India, Moths of Thailand, Moths of Singapore, or Moths of Malaysia. Okay, a few more minutes. In summary then, moths are an extremely diverse group. Observations you can make, doesn't matter whether it's daytime or nighttime, pretty much any habitat, anywhere, whether it's an urban park, national forest reserve, will have moths in it you can find somehow. By recording, record the methods, learn how to look, what to look for, learn how to keep that data both on INAT and in a secure backup environment so that you're contributing to the wider environment, the wider community that makes sense of that data for the benefit of everybody and everything. We only have one planet, after all, that supports life. So, best position, point of view to take photographs that illustrate the characters on the head, in particular, for your best chance of an ID. Don't forget the posture, wing and body shape. A side view, please, especially for the micros. Then worry about pattern elements. Actually, that's one of the last things to consider. And the last thing I want to emphasize is please do not assume that AI will be able to identify from your photo. Not everyone, in fact, very few people, even the specialists, have much hope of identifying if you don't show the features that I've gone through. Don't build up your expectations on that. Moth ID is difficult. It's why there aren't many people studying moths, I'm afraid. But the more that study, the more we understand, the more we learn. I hope this has helped you understand that. Keep being curious. Be diligent. Persist. I hope I've enthused you a little bit to go out and search, make life a little bit better for everybody. Right. Thank you very much. Say a big thank you to Asia Nature Challenge. And this was derived from various predecessory presentations from National Moth Week, City Nature Challenge, and originally Asian Lepidopterist Conservation Symposium. This is a 20-year program in the making. Off you go. Questions, please.
Yes, thank you, Roger, for that fantastic session. I mean, it was so gripping. Even I was also like, definitely this is going to help a lot of people. I'm really glad that you came over and gave us an idea on how basic classification and how to record moths, which is actually quite needed in these days. So I hope people will sign up the Asian Nature Challenge as a participant and do focus on moths as well. Let's document a lot of moths in across Asia. So looking for the questions, we got a lot of questions again. So one of the question was with respect to micromoths. A lot of people really want to know that how do you sedate micromoths, euthanize and preserve it, especially in humid conditions, because freezing usually gets results in damaging of the specimens some of the sampling techniques for micromoths, all those kind of questions. So in the similar area.
Take yourself a cold bag into the field if you're going to be doing collecting of any specimens, whether it be micros or macros. Select a container that is big enough for the moth to fly around in without bashing itself on the container, and pop it straight into the cold bag to stop it from flying around. Those really are the main factors that will give you a good quality voucher specimen in the long run. Setting material, there's bucket loads of information out there on how to curate a good specimen. But the really important bit is making sure that what you get in the field looks as good on the setting board and in the museum curated trays as it did when it was alive. And you've got to stop the thing from flying around. That's the main thing, and that's best done in a cold environment.
Right. So someone asked that so when considering about temperature changing and rainfall, humidity like factors, also showing influences for the visiting rate. So they are asking how do these environmental factors are affecting the moths, or is there anything special about it?
Oh, there's lots and lots and lots more. There are volumes of papers you could bind together that would fill a whole shelf. Literally hundreds of papers on environmental factors impacting activity of moths, and those are the key phrases. So go online, search. But yes, environmental factors enormously impact what moths are active.
Yeah, that answers.
We haven't got time to go deeper. It's just a big subject.
Yeah, agreed. Okay, so I saw two, three questions on the same area that like how is the moths basically influenced at the light pollution, and how is it like affecting their population in general?
How long have we got? This is like
I
that's
mean, we
another
are fine with it. It's up to you.
big subject. One of my collaborators is in the throes of finalizing a project on this subject at the moment. But if you look at the work by Douglas Boys in let's say 2015 through 2021, what they found in the UK is that yes, lights do seriously impact upon where moths are distributed and how they go about their daily stroke nightly routine. The work that we've been doing in Hong Kong has shown that areas that are already severely impacted by lights, the moths might actually be adapting. So the jury isn't yet back on the full impacts. Suffice to say, initially, definitely exceedingly negative, and it will deplete moth populations dramatically. But it is only one factor of many that are causing overall declines globally in moth and other insect populations.
Okay, right. And maybe a last question for today.
Okay.
Can you touch upon that topic wherein we say that moths are ecological indicators and they indicate some certain environmental factors happening?
Right. What I will do first is refer people to Roger Kitching's paper from year 2000, where they looked at assemblages of various moths in various families and how they were distributed in different habitats. And they came up with a generalization that seems to hold true for where geometridae are found and where noctuoidea are found. And one tends to be found in more open environments, one in more forest, and particularly healthy forest environments. That is one example. You can also look at, for example, and I've co-authored a paper on this, the footman moths, Lithosine, Lithosini, aka lichen moths. Now, lichen, some people will know, is sensitive to sulfur dioxide pollution. But lichen studying is even more taxonomically challenging than studying moths. So using moths as a proxy for lichen presence, absence, abundance, biodiversity, you can say that the degree of pollution impacting upon lichens can be measured using lichen feeding moths. Those are two examples. There are other examples for water pollution levels, with the accentropony in cranberry, for example. If you look at habitat, the way habitat is structured, compare grasslands to shrublands, to meadows, to forest. And in 3 dimensions, those are very different structural habitats. Now, people who are birders, for example, will know the differences in adaptation for habitat between hawks and falcons. Falcons have long, narrow, elongate wings for being in an open environment, whereas hawks are forest birds and they have broad wings for maneuverability in a structurally complex environment. Moths are exactly the same. Forest moths are broad-winged, lightweight, maneuverable moths. Open habitat moths tend to be long-winged, slender-winged. Lots of noctuidae, lots of hawk moths, for example, are in more open habitats. So there are a number of ways of looking at how the body structure indicates the habitat usage of a moth, or biomoth. Does that help?
Yeah, that gives a very good insight into it. I guess so.
Right.
We
Well,
have any
I'm
more questions?
sorry, I've
You,
run over a little bit on
okay,
my
okay,
2
okay.
hours,
Sure,
and
sure,
I'm
okay.
well overdue for dinner. So what I'm going to suggest is, if people still have questions, post them to INAT, to me on my INAT account. Don't worry, I don't mind receiving messages as long as it's relevant. What we can probably do is summarize those other questions on the Asia Moth Recording Program or on the Asia Nature Challenge project on INAT. How does that sound?
Yes, or you guys can ping me or put in the WhatsApp groups. I'll definitely get it sorted with Roger.
Okay, so everyone else out there, go through WhatsApp on Ashwin through the Asian Nature Challenge WhatsApp, and then we follow it up on iNaturalist over the City Nature Challenge week and beyond, quite likely. Then all that remains is for me to say thank you very much, Ashwin, for being host. Thanks to everybody who's tuned in at whatever point in time during the presentation. I must apologize for overrunning by quite a lot, but I hope you'll have gained a bit more insight in the overrun. Thank you very much. I'm signing off. Take care.
Thank you, Roger, for joining. Thank you, everyone.